Hi, I'm Gage Kinwissing. I'm here with Joseph Avenoso and Adam Elwood. We're going to talk to you about our experiment called The Effects of Microgravity on Osteoblastic Bone Reformation. Unfortunately, we had three other members of our group, Alex Belli, Rotem Hertzberg, and Tejas Patel, who were unable to attend. So to start off, I'll talk about the motivation behind our experiment. As it's been mentioned earlier in this conference, but the loss of bone mass in space for astronauts is a very serious issue. In fact, astronauts can lose approximately 1.6% of their bone mineral density per month while in microgravity environments. To put that in perspective, that's about 10 times the rate of bone loss of a woman in most postmenopausal osteoporosis. So it is a very serious problem in microgravity. And the reason that happens is that the human body is obviously accustomed to being in an environment with gravity. And when you're exposed to a microgravity environment, your brain realizes that it doesn't need the bone structure or the bone mass to hold the body together. So your brain will stop maintaining it and will actually start removing some of the calcium from your bones, making them much weaker. And to recover from this bone loss, you need about twice as much time as you spent in space to recover on Earth. So if you were to spend 90 days in space, you need about 180 days to recover back on Earth. And if you see in this picture, on the left is an hit the wrong button. On the left is an osteoporotic bone. You see there's a lot of the matrix broken down. It's a very porous and weak bone compared to a normal bone on the right. And that's kind of the extreme of what happens, but that's what happens when you lose bone mass. That's what happens to astronauts in space. And to put kind of a perspective on the type of cells that we used, this is the process for a bone being repaired by your body. So this isn't a huge fracture, but it would be a normal repair made when you have like a micro crack in your bone. So if you see on the left, the black line there is a micro crack. And the first step is that the osteoclast cells, the green cells, come in and they clean out all of the dead bone. Followed by the osteoclast or the osteoblast cells. These are the cells that we used in our experiment. They're the cells that actually come in, recreate the bone matrix, and create the new calcium matrix around themselves.
All right, so we know that it's the brain that makes the bones grow in our bodies. However, is it a possibility that these bones or that these bone cells can grow in space at all? Well, that's what we really wanted to find out. So to run this kind of experiment, you need three things. You need cells, you need something for the cells to live off of, and you need means for the cells to grow because we can't exactly bring a whole brain up into space. So what we did was we took a colony of embryonic mouse osteoblast cells, which if you guys can recall, these are the cells that do end up forming that bone matrix. We used a DM-EM nutrient broth, which was approved by NASA, and then we also used a human growth hormone. Now, some of you are probably thinking, they used a human growth hormone on cells that came from a mouse. Well, we knew this would actually work. This is a once-in-a-lifetime opportunity. We weren't just going to do this. So we found research from France from the 1990s in which they used these same cells and they used a human growth hormone. So this has worked before, and therefore it did work for us, at least with our control group that was monitored at UMD&J. These are very delicate cells, so we were not able to work with them by ourselves. We would have killed them very easily. They're very sensitive. So, of course, what we had to do was we had two vials. In one, we had the cells in the nutrient broth, and then we also had the human growth hormone in the other. So once they were up in space, a day later, the two vials came together and they mixed. So then the cells could have the human growth hormone so that they could grow. Soon after, they exhibited what is called confluence, which is when they form that bone matrix. If you guys remember that the cross-section of the bone, when all the white crisscrossed bone, they come together like that, that's what we wanted to see in the bone. That's how they grow. They don't exactly grow bigger. They just form that matrix and make the bone stronger. And then about 10 days later, the control cells on Earth began to actually expire and die. And then when we got the experiments back from the cells back from space, we noticed a similar timeline.
These are actual pictures of the bone cells that we have collected. They're the bone cells collected at UMD&J on the launch date of the shuttle. So, our experimental results. As Joe said earlier, we did receive similar results on Earth that we did get from space. What we did to look for our actual results was look for confluence, amount, differentiation. So confluence, as Joe said again, was the matrix structure that builds. It's kind of like a lattice formation inside the bone. We did see that starting to form in each vial. The amount of bones that started to show this confluence, and the differentiation from osteoblasts into mature osteocytes. Unfortunately, this study couldn't be done really in depth until we had to bring the cells back from death. So we had to resuscitate the cells using a replenished amount of oxygen and nutrients that we put into each vial, both the one from UMDNJ on Earth and the one from space. So after we resuscitated the cells, we waited a while and then we conducted the study. What we did learn, although the results were inconclusive, we did learn that these very finicky, very delicate cells, the osteoblasts, could grow in space, and there's a lot of promising work in the future that can be done. Also, prior work from NASA has actually shown that this similar experiment has been conducted on chicken osteoblasts, where, once again, they received similar results. I would also like to thank specifically Sigma Aldrich and the European Collection of Cell Cultures for donating all of our supplies. I'd like to thank Dr. Chris Britton at UMDNJ for helping us and letting us use his laboratory. I'd like to thank friends, families, collaborators, the Bridgewater community, and Dr. Valdez. Thank you very much. We'll now take questions.
All right. Thank you for an excellent presentation. Questions for this student team, please come up and ask your questions using the microphone. Thank you.
Hello. I was wondering if you can please explain again how you resuscitated the cells?
We took the old cells, oh, that's too close. We took the old cells from the vials, both in space and the ones, the control groups on Earth, and then we basically replenished their oxygen supply by dissolving it into the nutrient broth. So we basically gave them food and air, which they were deprived of in space.
Thank you.
You said the life expectancy of ones in space was about 10 days. How can you tell on those dead cells that they just lived 10 days like they did about on Earth?
We can't. We weren't actually in space to see the cells die, so we assumed that because they were in the same type of environment that was controlled on Earth, that they would have died as well in space. So, yeah, we did get the same results, and when they came back, they were dead, so it happened sometime in there. All right, thank you very much.